So my name is Ari Kahn. I'm with the Texas Advanced Computing Center. I'm part of the Data Integration and Resource Center of A2CPS. I'm also the PI for the Data Coordinating Center. Um, and I want to tell you a little bit about how the data flows within A2CPS. So up here in the top left, we have the clinical sites. And what they're doing, all those kind of data that Martin was talking about, it's entered into REDCap, whether it's clinical data, subjects, new subjects, if they're creating or doing blood draws and then shipping those off, if brain images are being collected, the psychosocial data, the PROs, those are all collected in REDCap. And and that's connected to our virtual biospecimen repository at the DCC, and I'll talk a little bit more about that. But when the biospecimens are collected, they're after they're collected, they're shipped off to a sample management group at University of California, San Diego, and they collect all the samples until there's enough to actually send off to the omics centers for analysis. And uh, once there are enough, they're shipped off to the omics centers, and we provide them with run lists, basically to optimize to minimize the batch effects that might happen, but also to optimize their assay flow, right? So we want to I guess minimize their expenses. And after they do their assays, they submit their results to the DCC here. And then the Data Integration Resource Center, the DIRC, we have access to that, and so that we can start analyzing it. So the virtual biospecimen repository, we're trying to figure out and keep track of all these samples because they're valuable. And so we want to know, like, what container they're in, which tube they're in, what box are they stored in, but where are they being shipped? Did they actually arrive at the destination? Who has them? And were there any exceptions? And is there anything left from the assay? Like I said, we also provide them run lists, and then when they submit, provide the ODGC's run lists, and then when the ODGCs submit data, or the clinical sites or the medical sites submit brain images, then we can link that data to the subjects and biospecimens that they're associated with. So we have this cool API. If we provide these slides, you can click on it and check it out. So this is how we process all the data from REDCap. So we actually have, and anytime there's some event, we get kind of a push webhook. And so depending on whatever the instrument is or whatever the form is, we have something to handle it. And the goal is to collect all that, to collect all that data into our database. And we're using Postgres with a API on top of it. Also, whenever there's a new event or new shipment of biospecimens, we get a notification so that we can track it. So we keep know what's going on. So our goals are to validate the data and then to link the data with all the other patient-based data. So the subjects, samples, the AHR data, psychosocial, MRIs, the omics data. And then as data is submitted, as the omic data or the imaging data is submitted, this is kind of showing a EHR centric one, but we have a data type specific handler. So whatever the data is that's being submitted, there's a specific handler. We know that a new file has been submitted, and we know what kind of file it is, and so we know how to handle it and how to validate it. And then the validated files are deposited, into directories, but we keep track of all that in our database. So data sharing. So due to the study of the design, we're not sharing anything except for the baseline data until the study is completed. So 6 months after each internal release, we're going to deposit the baseline data or the pre-surgery data into the NIMH data archive, or the NDA. So as Martin said, the first deposit is expected this fall. And then 1 to 2 months after each NDA release, right, we're also going to submit the metadata to the CFDE so that you can actually use the CFDE portal to find those data and relate them. And then 6 months after the study is completed, all the remaining data is submitted to the NDA, and then that includes the rest, the whole of all the data that we've collected. The metadata for those submissions to the NDA is also submitted to HEAL, and also to that, I didn't put CFDE here, but there too. And then if there are any leftover biospecimens, there's actually a HEAL biospecimen repository. And so we'll collect all those biospecimens and ship them over there. Okay. All right. So these are all the major data types and where they are being deposited. Basically, everything gets deposited into the NDA. Except for the project metadata, which gets submitted to CFD and HEAL, and the leftover biospecimens. This is this is how you get to the NDA. It's I think the NDA, a lot of the data that's or a lot of the data structures and the kind of the shape of the data is kind of being developed as the NDA progresses and as those new data get submitted. But it's a highly curated data set, which means that the metadata surrounding it has very specific data structures that we have to submit to. So this link here will go to all the harmonization standards that they have, but we can go into some of it if there's time. But basically, there is controlled access and open access data available there. One interesting aspect of the NDA is that they have these computed subject GUIDs. And the idea is that you have this base set of metadata about a subject that you put into a program, and it spits out a GUID that's specifically associated with that metadata. So it kind of obfuscates the metadata, but it provides a consistent ID so that you can find the same if that subject is part of many different studies, that you'll find that same GUID for that same subject. That makes sense. One thing that's important to know is that the NDA is on Amazon Web Services, so the download to non AWS is limited, but if you have an AWS instance of any kind, then it's unlimited data transfer. And so this is kind of what Martin was talking about in terms of like how the data kind of hangs off of time points. So you either have, one of two cohorts, the total knee arthroplasty or knee replacement or the thoracic cavity, and then you have a patient or a subject, and then you have EHR data about that, and then you have time points. So the time points are either baseline or these other ones, 6 weeks, 3 months, 6 months after surgery. And all the data kind of, hangs off of there. And these are the data that you'll have access to until the end of the study. How much time do we have?
About half an hour left, Ari. We have a
Okay.
question. Is now an okay time to pause for questions since we
Yeah,
paused?
let's do that.
Sure. So the question was from online. It's for older adults who often go post-acute rehabilitation locations outside the hospital EHR and who undergo interventions and take medications that could affect pain. Does A2CPS acquire these important data points?
I can answer that question. Yes, we do. At each time point, we ask about sort of different treatments, that's been done since the last visit, and so and that includes both medication and seeing other professionals. It could be sort of like acupuncture or whatnot. Yeah. So yes, we do collect that.
Beautiful. Thanks, Martin. All right, Ari, how you doing? We have staff
Good.
back to you. I don't have too many other things, so I think you're good to go for a few more minutes.
Okay, well, I don't know if I want to if we want to go into it, but I do have slides kind of like on each data type, but I guess I can go through it real quick. The clinical and psychosocial data, so there is some controlled access data, but we're basically anonymizing all of it. And so there'll be open access versions available. Again, it goes to the NDA, and the metadata standards are the NIH HEAL common data elements. And then we have EHR data, which Seth is going to talk about. And they can be open access, and we link all the data to that's associated with a subject to the EHR data. The CDEs for the EHR data are going to be in the NDA. With imaging data, so it turns out that like a full image, you can reconstruct a face, and also the DICOM files have PHR, PII data embedded in them. So what we do is we create these skull stripped Nifty files, and those can be open access. So those are submitted to the NDA, but we might submit them to other repositories as well. And we use the ABC metadata standards. For gene variants, those are controlled access. They're basically a SNP chip, and so the file types are IDAT and VCF. XRNA, those are open access, and if you're familiar with XRNA, you have FASTQ files and counts. For proteomics, we actually have two groups that are doing proteomics. One of them is a Luminex-based platform, and it's an immunoassay, and this is a primary biomarker. There's also a Thermo Fisher platform, and it's a spectroscopy. And so the file types are, they have a raw file or an MZ file, and then there's these other processed outputs. Metabolomics and lipidomics, they don't contain any protected data, and you kind of have these set of data that are specific to those kinds of platforms. And then there's the project metadata that I talked about that are being submitted to the CFD and HEAL. And it's using the for the CFDE, we actually based our database off the C2M2 model that the CFDE uses. And so if we can submit it when we're ready, we can just submit it because it's already ready. And then the metadata standard for HEAL is the HEAL CDEs. And that is all. Seth, wait, are there any questions?
That was great. Ari, when should users from the external perspective start to see some of these data rolling off from you as being accessible to the community? Or did you already cover that?
Great question. So they should be available at the beginning of 2025.
Okay. Cool.
I think it's fair to set your expectations because they'll be submitted in the fall, but then there's like Thanksgiving and Christmas, So I think that's probably.
Beautiful.
All right.
Awesome. Thank you, Ari. That was great.